Hi, everyone. Happy to have you here again for our bite-sized talk today. We're joined today by Nicolas Servant, who's based at the Institut Curie in Paris, France, talking to us about the nfcore HiC pipeline. If you have any questions for Nicolas during the talk, please put them in the chat or even unmute yourself at the end of the session and ask them directly. So we've started doing that from today's talk. And just to let you know, this video and all previous bite-sized talks are on our YouTube channel. So you can feel free to consult them there as well. And all links are still on our homepage. So with that, thanks for joining us again, Nicolas. Over to you.
Thank you very much. It's a pleasure to be here today to discuss with you about the nfcore HiC pipeline. So for today, I'm going to first introduce a little bit the project and the context about the chromatin organization. Then I will describe the main step of a typical HiC data analysis. Moving to the nfcore HiC pipeline, I will show you how to use it and how to look at the results. And finally, we will discuss a bit the future developments of this pipeline. Okay, so very broad introduction and very simple introduction to the scientific context. So we are here interested in the field of chromatin organization. And what we know so far is that you have different levels of organization within the nucleus. So the first one is about chromosome territories, which has been described quite a long time ago using microscopy, where actually we know that you have each chromosome occupy its own space within the nucleus that we call chromosome territories. Then if you zoom in at the level of a chromosome, a couple of years ago, it has been described that you have broadly two main types of chromosome compartments, where on one side you have all the chromatin which is active, open, with transcription and regulation. And on the other side, you have the chromatin which is inactive, much more compacted, which is, for instance, enriched in repressive histone mark and so on. So we know that you have other subtypes of chromatin compartment, but for today, we will just focus on A and B, active and inactive. Then if you zoom in again at the level of the megabase scale for mammals, we have described a couple of years ago what we call TADs, or topological associated domains, which are kind of subunit of regulation, where within a topological domains, you have all the regulatory elements that can contact genes and promote transcription, for instance. However, you have almost no contact between TADs, because in the middle, you have a kind of barrier, which are made by insulation protein like CTCF coenzyme. And if you zoom again at the very final scale, you can start to see loops between regulatory element and genes, for instance, with very high resolution data. So you have this four level of chromatin organization, and you can imagine around that a lot of different biological questions. So how does it change in different conditions during differentiation, I don't know, embryogenesis or whatever? What will be the impact and the link between gene regulation and chromatin organizations? For instance, you have now in the literature a couple of very nice studies that have shown that if you disrupt the topological associated domains, you may have some non-expected gene regulation and so on. So that's for the context. And how can we have access to this information? In 2009, Lieberman, Hayden, and Baker's lab proposed what they call the HiC Protocol, which is a genome-wide 3C approach. So very briefly, here you have the main steps of the protocol, where actually you're crossing the DNA, then you cut using a restriction enzyme or something else, you ligate, you pull down the ligation products, and then you sequence using parent sequencing. So at the end, you have this kind of maps that we call contact maps that you can look at different resolutions. So here you have the chromosome, the genome-wide maps, where each square here is a chromosome. You can zoom in up to a given resolution. And of course, the higher is the resolution, the higher should be the sequencing depth, up to almost 1 billion reads per sample. So if I try to make the link between HiC and the different level of chromatin organization that I showed you before, you have the chromosome territories that you can see here on the very small HiC maps where you have just 4 chromosomes. And you do see that each chromosome has contacts with itself, but not that much with other chromosomes. Then if you look at the level of a chromosome map, you have this kind of checkerboard pattern, which is highly correlated with the AB open-closed compartments. If you zoom again, you start to see what we call the TADs, which are this kind of triangle around the diagonal, up to direct loop and contacts between loci and the genome. So to come back to the protocol, so you have the fixation, digestion, your ligate, your enrich, and your sequence. One thing which is important to know when you want to analyze such data is that for a couple of years now, we had quite a lot of different protocols that were proposed for HiC data. For instance, in 2014, we have the in-situ HiC, then the single-cell HiC. You can also change a little bit the digestion step using DNase, for instance. We do have a couple of kits which are available on the market now. You can also enrich for a couple of targets at the enrichment level using capture HiC, or even combine the HiC with kind of ChIP-seq-like protocol to immunoprecipitate your protein of interest. This is what we call HiChIP. So you have many different protocols which are derived from the initial HiC protocol. Okay, if we move to HiC data analysis, again, it's quite very simple, but you have 3 main steps in the analysis. The first one is the reads mapping. So where actually you do have this kind of parent sequencing, where you have the blue part, which is from the first interactor, and the red part, which is for the second interactor. And you just want to align those reads on the reference genome. A few tips about the HiC mapping is that first you may have to deal with what we call chimeric reads, especially, for instance, if you have one reads spanning the ligation junctions here, meaning that on the single reads you will have the 2 interactors. So usually what people are doing is that you just want to map the 5' end of the reads. And the other thing which is important is that you don't want to put any constraints on the distance between the 2 reads, because you can have short or quite long-range contacts. So what you can do, for instance, is to align the first read on one side, the other reads, the R2 reads on the other side, and then merge the alignment to avoid any constraint based on intersize. Then once you have the reads aligned on the genome, there is a huge step of filtering, where actually you want to keep only what we call valid pairs, which are the valid 3C products, so the valid ligation product that you have on your data. So here, this is an example based on digestion HiC protocol, where actually you will check where on which restriction fragment each read is aligned, and you will try to reconstruct the ligation product. You have a couple of read pairs that you will discard, such as the one singleton, so the one for which you don't have the other contacts. Dangling ends, meaning that actually there were no ligation here, but so you just sequence one fragment which was alone on the prep. Self-cycle, I mean ligation against itself, or any other product that you cannot really explain. And of course, you can add any additional filters based on mapping quality, insert size, duplicates, and so on, like restriction fragment size, and so on. So once you have detected only the valid pairs, you can start constructing what we call the contact map. And to do so, that's not very complicated. You have the first interactor A and the second interactor B, and you will just count how many times A and B has been sequenced together to try to estimate contact probability. So what is the probability that 2 pieces, 2 loci in the genome will interact together? There is an additional step, which is about normalization. And here we're not entering into the details, but you have a couple of algorithms which are called balancing algorithm that can be applied on the HiC maps. If you want to have details, I can suggest you to have a look at the Imaka et al. Paper in 2012. Okay, last thing about the HiC data format. So in the field, you have three main types of format for HiC file. So the first one is a text file, which is pretty simple. Basically, because HiC maps are symmetrical, you only store half of the data. And because you also have a lot of zeros on those data, so you can use a sparse format to reduce the size of the text file. Then in the context of the 4D nucleome, they introduced two other type of HiC data format, the HiC files, which are derived from the Juicer suite, which is another workflow for HiC data analysis developed by the Lieberman Agents Lab, that you can put into Juicebox, which is a tool for visualization of HiC map, or now for a couple of months into the UCSC Genome Border. The other one is the cool file, which really tends to be the standard format for HiC data, because it's Python oriented, so you can easily load this file into Python and do the analysis by yourself. There are many Python packages that can handle these files, and they are compatible with HiGlass for HiC data visualization. So one word about about downstream analysis. So basically, once you have the contact map and you want to go further, so looking at the topological associative domain, the loops, and so on. So here, to my knowledge, there is no standard yet in the field. And to illustrate that, I present here a picture from the Forcato et al. Paper, where actually they try many different algorithms for task calling and for loops calling. And you can see that you have many different results. So I will come back on that later, but in the context of a standard pipeline, that's not that easy to choose one tool among the others. But it's maybe better to propose different tools for that. Okay, so moving to the nfcore HiC pipeline. So we started to develop this tool in 2019 now, so 2 years ago, and we are now at the version 1.3. So the main why we did that, it's really to have a community pipeline. So everyone is very welcome to contribute to this tool. And the idea was to have something which was able to run the data processing, to run a couple of quality control on the HiC data, to check all the steps of the protocol, and also to start moving into downstream analysis. I told you before that you have many different protocols, and the idea was to be able to support all this protocol in a single pipeline. And finally, one thing which is also maybe one of the most important one is to follow the nfcore good practices and to have something which is reproducible, which is easy to use, that you can use on the cloud, on your own machine, with all the containers that you like to use, like Docker, Singularity, Conda, and so on. So the first version of the pipeline was clearly a kind of Nextflow based implementation of the HiC pipeline that I developed quite a long time ago, which is still highly used in the field. So HiC Pro start from the FastQ file, perform the reads mapping, detect the valid interaction products, and construct the maps. So the first version of the pipeline was really taking the HiC Pro pipeline and building a new one using Nextflow and the nfcore good practices. Then from version 1.3, we started to move a little bit. And especially for the contact maps, we are now using the cool files, so the cool format with the cooler package. You still have option for backward compatibility if you want to have exactly the HiC Pro maps. That's totally feasible. And then we also started to implement a couple of tools for downstream analysis to call the compartments, to call the TADs using two different methods. So either the insulation score or another method, which is in the HiC Explorer package, and also to add a couple of additional QCs like distance decay and so on. So how to run the pipeline? There is nothing very complicated compared to the other nfcore pipeline. You just have to provide the FastQ file and the genome of reference you would like to work on. And if this is a digestion protocol, you just have to put the name of the restriction enzyme that has been used. And automatically the pipeline will set up the restriction site or the ligation site that will be used for the analysis. So in the first version, you had to set up these two options manually. Now it's almost automatic. And if for any reason you are using an enzyme which is not in the list, you can still set up these options manually. And we also have a mode for DNase IC. So if you don't have any digestion, but if you are using DNase instead of restriction enzyme, and in this case, you just have to use the options DNase. And to filter out all the invalid pairs, we are using the minimum distance between the two contacts that you have to set up also. So this was the very simple command lines to use the pipeline. Here I'm giving a more advanced command line to give you an example of what we can do. So if I have to translate this command, basically it's the same. I'm giving my FASTQ file. I want to align on NG38. This is a digestion HiC, so made with DPN2 enzyme. I told you before that actually HiC can be very highly sequenced, very deep sequenced, up to 1 billion reads per sample. So it's very useful to be able to split the reads into chunks and to analyze all the chunks in parallel on a cluster. So to do that, we have an option to split the FASTQ, and you set up just the number of reads that you want per chunks. So here I put 10 million, yeah, 10 million reads per chunk. And the FASTQ file will be split and analyzed in parallel on the cluster. Then I ask the pipeline to generate the contact maps at different resolution, 20 KB, 40 KB, 250 KB, and 1 megabase. So these are the size of the window we want to have on the maps. I want to call the compartments using the 250 KB resolution. And then I want to run two TATS caller, the insulation and HiC Explorer using a resolution of 40 KB. So this was just an example of a more complex advanced command line. So at the end of the pipeline, you do have a couple of quality controls, which are so far based on the HiC Pro module that you have in multi-QC, where you have a couple of graphs. So the first one is about mapping statistics. So how many reads are aligned on the genome. You have in dark blue the reads which are fully aligned, and in light blue the one for which HiC Pro found the ligation sites. So these are the reads spanning the ligation sites. So we trim the reads and we just align the 5' ends. So this can already give you a good information about the quality. I mean, the quality of the data. Do I have ligation motif in my data set, which is of course expected. And then we reconstruct the pair and BAM. So we merge the two R1 and R2 BAM files. And here again, you can filter the data according to the mapping quality and so on. Another very interesting quality control is about the valid pairs detections. So you will expect to have a lot of valid pairs, much more than invalid pairs, but you have the details about the different type of valid pairs and the different type of non-valid pairs that you have on the data. So this is very useful if you have issue with the ligation step and the protocol, with the pull down step. You can try to understand what's going on and to fix your protocol after that. And finally, last plots, you have the representation of the distance between the contacts. So you have a lot of contacts in cis, so very close contacts, which is expected. A couple of contacts in trans, so between chromosome. So that's also very interesting to look at that. Okay, so about the future developments. So the most important one is to be able to port the pipeline into DSL2, which is something that we have to do, I hope, this year. Then there is many things to do. Adding other mapping tools instead of HiC Pro, if anyone is interesting. Being able to merge multiple sequencing lane, especially for very high sequencing depth. We also think about adding a couple of additional QCs in the multi-QC report and propose other loops calling tools, stat callings, and so on. And the last one, I would like also to move on capture HiC protocol, which is a bit different, but not that much compared to the HiC standard protocols. And of course, contributions are very welcome if you want to participate to the development of this pipeline. To conclude, yes, I think that's it. Here I put just the link of the project, and I really want to thank the nfcore and all the community, because that's really a great project, and I'm really happy to be part of it. Thank you very much, and if you have questions, I'll be happy to answer.
Thanks a lot, Nicolas. That was a very, very clear talk. Are there any questions? If you have any questions, please put them in the chat, or you should be able to unmute yourself and ask them directly. So please go ahead and do that.
Hi, Nicolas. That was a brilliant talk. That was really interesting. Thanks for that. This is Phil here. I was just thinking about, you were talking about the HiC protocols doing chip-seq on the HiC products. And then also saying at the end about you'd like to migrate to DSL2. I'm just wondering if there's any synergies between the existing chip-seq pipeline and whether we can sort of share any of the underlying modules and maybe even sub workflows there. Could you elaborate on what that type of analysis looks like?
Yeah, so about the HiC, basically all the first steps of the analysis is almost the same. So you have no difference. The only thing is that you will enrich for a couple of ligation products that are really focused on your protein of interest. Then, however, when you want to go forward, looking at, for instance, detecting loops or really a specific pixel on your contact maps, you have a couple of tools which are dedicated to HiC. So indeed, that's something that we can add. And as you say, the fact that you are using DSL2 will really ease with a kind of sub workflow dedicated to HiC data analysis.
Nice. As one other thing as well, you mentioned about merging sample lanes at the end. Hopefully, most of the pipelines moving to DSL2 are also switching to using sample sheets to take their inputs instead of doing a file glob. So I guess that will be a nice solution for that.
Yeah, absolutely. I saw that on the Slack. I have to move on that. You're right.
And we have a question in the chat from Gabriele, who thanks you for a very nice presentation, first of all, and then says that they have a kind of technical question. Specifically, they're wondering about the advantages of HiC over Cool or vice versa, and about other formats like FANCI or PairSam.
So basically all this, so yeah, for me, the main advantage of the Cool file is that first you can load them into tools to visualize the data. So there is this eyeglass server, which is a very nice and powerful tool to easily zoom in the data. You can zoom out. You can put different files in the different sample together and compare them. So that's really powerful. And the other thing is that because the Cool format is really Python driven, it's pretty simple to load the data in the Python framework. And so you have many additional tools that are able to start with the Cool file. Then compared to the others format, well, I think that's more a matter of philosophy. I don't think that there is a lot of differences. All these formats are now highly are able to store multiple maps at different resolution, and they do not take a lot of space on disk, which is also something which is pretty interesting. So on the community, we do see that the Cool file seems to be one of the standard that you can, for instance, also put on Geo on the CBI database if you want to share HiC data between teams. Yeah, that's it. I think that's better than text file because it's also smaller.
Thank you. Does that answer your question, Gabrielle, or do you have a follow-up question? Okay.
But yeah, you also have now a couple of tools which are able to start from one file format and to transfer in another one. So that's not a big issue so far.
Okay, so I think that answered his question. He's quite happy about that. There are no more questions that have popped up so far. I'll just take a final look at YouTube.
One, sorry, quick naive one while you're looking, Renika. Is the analysis of DNA's HiC the same as MNA's HiC?
Yeah, absolutely. So the only thing which change is the way you will digest the fragments. So you can use DNAs, you can use MNAs, you can use micrococcus and so on. However, the main limitation is that when you are using transcription enzyme, basically then you can build a kind of in silico reference of all the different restriction fragments, because you know exactly where the enzyme is expected to cut, which is not the case for the DNAs, for instance, or MNAs, whatever. So it means that for the DNAs and MNAs protocol, it's a bit more complicated to be able to call the, for instance, the dangling ends, because you don't know exactly where where you have cut on the protocol. So that's why there is nothing special. We are just using the size, the distance between the two reads, under the assumption that if the contacts are very close to each other, it's likely to not be a real ligation product, but more just one fragment that you have sequenced alone. But yes, yeah. So there is nothing. I mean, yes, that's the same for you. And but I'm pretty sure that you have a couple of maybe in the pipeline we can add a couple of additional steps to better analyze this kind of DNase HiC data.
Okay, so then there's a follow-up question there about from Gabriele again about whether the nfcore HiC pipeline supports multiple restriction enzyme HiC.
Yes, absolutely. So I didn't mention that, but you can of course have multiple enzymes. I think that's the case for the commercial kits that are recent on the market. You have multiple enzymes, so you just have to. I have to check, but you just have to specify the enzymes, comma separated, something like this. It should work.
Great. So there are no more questions at this point. But you know where you can find Nicolas. You can contact him on Slack and ask questions also on the bytesize channel. And thank you again, Nicolas. Thanks for taking the time to give this presentation to us today. And I'd also like to thank you all for joining and for the engaging discussion after, and the Chan Zuckerberg Initiative for funding. So we'll be back next week with a talk on how to write pipelines to DSL2 by Harshal Patel. So join us again next week at 1 p.m. CEST. Have a nice week, everyone.
Thank you.